Uh, I'm Susan Sullivan. I'm director of the Series Education and Outreach Group. I'm the MC, for lack of a better word, for this event today. And before we get started, I want to acknowledge the people who are part of the project, who have been organizing the project, some of whom are here today and some of whom aren't. So this is an NSF-funded project. It's part of the Research Experiences for Undergraduates project, and it's also supported by the Critical Zone Observatories. So that's been a fantastic partnership to get people in the field working on interesting questions. The leaders of the project include Leslie Smith and Anna Gold from Series, and Suzanne Anderson from InStar. Becca Batchelor is here, who's been running the skills workshop, the communication training. Jennifer Taylor is here. She's been managing, coordinating the project, and working with students throughout. I also want to acknowledge there's some of the mentors in the room. If you're a mentor, would you raise your hand, please? Yeah, great. Yeah, you can raise your hand. I know you're a mentor. We are very pleased with the project, and every year we're so proud of what students have been able to do. It's really striking how much people have grown and learned how to how to do science, how to make observations, how to analyze data, and very importantly, how to communicate your findings in the same ways that scientists do and that you do now as a budding scientist. We're always so pleased with the fact that we consider this to be a bridging program between your experiences in community college and whatever else you might do in your next step. And so we hope that this experience has given you a sense of how it is to work in science, and that provides some sort of information or clarity to help you know, do I want to keep going this direction? Is this interesting, but I want to go in a different direction? All of those are good outcomes. So we're very happy to have you here, and we're very pleased to be able to build relationships with you and your institutions. We also want to thank the mentors very much for your generosity with your time and your expertise, because we know that often you're building relationships that can have a lasting effect. We're going to go into the presentations, and so the format of that is that each student has 10 minutes to present and have questions. So you'll have an alert from Jennifer when you're into it and when there's time for questions, and then we'll transition in between. So questions, please hold any questions until after the presentation, in between presentations, and then we'll take those questions. We also are pleased to have this being on a webinar right now, so it's possible that we may get some questions from people who aren't in the room, who are with us virtually. And if that's the case, then Katie in the back, thank you for running the webinar. Katie will let me know if I need to interrupt and take a question virtually. So the first set of presentations kind of work together. And so the first three people are Randall, Rouse, Jesse Barber, and Brett Lindgren. And Randall will introduce the set of three presentations, and then each person will give their own presentation. So Randall, I'll let you do that, and then I will introduce your presentation.
Oh, well, basically the common factor that we all have is leaf area index. So for the next 30 minutes, you'll probably hear that term a lot. So my presentation is on evaluating the effects that topographical wetness index and slope orientation have on the leaf area index and tree height in coniferous forests. Jesse Barber did his on the correlation between the ground data that we took for leaf area index and the aerial LiDAR. And Brett, last name's escaping me. Perfect. He did his with solar radiation absorption through the canopy, which has implications with leaf area index as well. Cool.
So our first presentation is Randall Rouse and his mentor. I think I have to speak into this for the his presentation is evaluating the effects that topographic wetness index and directional orientation have on tree height and leaf area. And his mentor was Theo Barnhart from Instar and CZO.
Yeah, so now it's been introduced a few times. So that's what I'm doing. Thanks to Theo for his guidance. Thanks to Jesse for being an awesome partner. Thanks to the whole REC faculty. So let's just jump in. I guess probably starting with defining these terms. Leaf area index is the measurement of green leaf area to ground area, unit per unit. Topographical wetness index is the contributing upslope factor divided by the slope gradient. Slope orientation is the compass direction in which the slope faces, and azimuth is the direction which water would flow down a hillside. So my predictions for this project were if you have a higher TWI value, it would encourage a higher tree growth and a denser leaf area index, and that south-facing slopes would have a higher leaf area index as well. And that's just from personal observations and a little background reading. That's where I drew those predictions. So some of the tools we used for the collection of the data, we used a rangefinder to measure the tree height and take the slope of the hills. We used a DBH tape to measure the diameter of the trees, a compass for figuring out the azimuth. We used GPS to actually find the locations and then to take the coordinates when we were there. We used GIS to download all of our data that we collected in the field. And somehow I left it off, but we used a hemispherical camera to take canopy pictures. So that's I just went over that. But when we were collecting data, we identified all the trees within the plot, and we took the measurements from 25 random trees. Cool. So once we had all of the data that we collected in the field, which was primarily what we did all summer, was collect the data. We processed the pictures that we took with the hemispherical photos through a program called GapLight Analyzer, which generated our LAI values. We would put all of the data in the spreadsheet, and then between the single independent and dependent variables. Here's just a picture of the GapLight Analyzer here. Skip that. So when I analyzed just one independent variable, which is its average TWI, it's actually kind of hard to see, and northness over here, versus the tree height of the entire forest, which I thought would have, I thought these independent variables would have a universal effect over all conifers. I found absolutely that was not the case. There was no correlation between the topographic wetness and tree height or the northness or tree height. I guess I should clarify, northness is just the azimuth. It's easier to chart out as northness. It's just the cosine of the azimuth. And here is also the northness and average TWI of the sites versus the leaf area index of the whole coniferous forest, which also generated no correlations. So after I didn't find any correlations within the entire forest, I decided to group sites by dominant species, which limited my data sets, but I got some better correlations here. With my northness versus tree height of subalpine firs, an R² value of 0.57, which was the highest correlation I got with a single variable. And here we have the LAIs versus northness and LAI versus topographical wetness with very small correlation patterns. So once the single variables didn't pan out well for correlations, I went to a multiple regression analysis where I added the variables, the TWI and the northness together and saw if they correlated any better. And I just focused on the subalpine firs here. So for the first multiple regression with the slope and northness, I got a marginally higher R² value. with a p-value that's pretty high too, showing that the probability of chance is pretty high. But the fact that there's a trend here is better than the single variables were. And as well with the slope and northness versus tree height. Also a higher R-squared value, but a probability of chance that's a little too high for it to be statistically significant. So I did the same thing here with TWI and northness versus the LAI and tree height. Instead of just slope, I used the TWI values. And there was also some correlation here. And again, a probability of chance that's a little too high for it to be significant. And so that was my findings. It didn't really work out as intended, but the data sets were very small with the tree-specific sites. For future research, if we did more sites that had the same species, the probability of chance would either go down or up, and it would either validate or invalidate that trend. So that would be my suggestion for future research. Yeah, I pretty much covered that there. So some acknowledgements. I'll leave that for a second. And any questions? Yeah.
You mentioned the within your class you randomly selected trees.
Mm-hmm.
So two questions: how large were your sample size?
Okay. Yeah.
How did you select your trees?
So the plots were 30 meters square, and we had to take 25 trees, so we would basically make quadrants and select 8 trees throughout each directional quadrant, and then 1 tree that was at the center stake is typically how we did it. Any other questions?
You mentioned sites that might have a more homogeneous single species tree. Are you aware of any sites like that, or where would you look for something like that?
Probably I would look on Google Earth, and if you could get any idea from the canopies from above you, or just typical conditions that would have that, elevation. I would just have to do background research, or do map out, one site. Just do a lot of sites in like one particular place that has a different slope gradients, like throughout the site.
Okay, so next we have Jesse Barber, and Jesse will describe the terrestrial-based leaf area index compared to leaf area index derived from remote sensing for the Boulder Creek Critical Zone Observatory.
Does this work like that? No. All right. So good afternoon. My name's Jesse. I'll be talking to you a little bit about ground-based leaf area index, or LAI, compared to remote sensing LAI. So what is leaf area index? Kind of like how Randall described it. It's the amount of leaf area above a certain ground area. So it goes from a value of 0 to anything really, depending on how tall the trees could grow, if they could just grow out to outer space. So, but realistically, it's like 0 to about 2.2 is kind of what we got for our highest value. And then the important factor, why it's important, environmental models for solar radiation, like Brett's going to talk about a little later, or gas exchange models, you need LAI in those. So the purpose of the project was to collect ground-based LAI because there wasn't a data set in the Boulder Creek area for ground-based LAI measurements. So that's kind of why we did it. And then my project was to compare it to the remote sensed LAI to see if there was a correlation or not. And then the goal was to get a good correlation, hopefully. So we'll see if we did. So locations where we did it, here's the Boulder Creek CZO outlined in this pink purplish color. All these triangles are sites we actually went and processed. So we did 41 sites, plus this is just a test site, so we didn't actually do one in the middle of Boulder. So, but we focused mainly on right here, which is Golden Gulch and the Mountain Research Station, because that's where a lot of projects are. So we wanted to make sure we had a good data set for those areas. And then, so this is how we laid out each site. We used the GPS to find it. And then, so that's the star is the center spot, which we found with the GPS unit. And then we use a tape measure, and we'd measure out so many meters away to get all these other sites. And then, so we would take, after we measured it all out, we would take photos. Here's the hemispheric camera that he mentioned earlier. We set that up over each stake, and we'd orient the top of the camera to be pointing northward. And then, so we'd take pictures there. And then, this is a GPS unit we used with a pole and an antenna to get the GPS points at each spot, so we knew exactly where we were at within a couple meters. And so we take all that data, all the pictures, we put it into our computers, and we use this program which Randall mentioned earlier. This is Gap Light Analyzer. And so the picture on your left is the original picture the camera took. After it's been processed, it goes to this black and white image. And so the program takes these black and white pixels and then compares them and gives us an LAI rating. So this picture was, I believe, a 0.75 rating, just for context. And then, so this is ArcGIS. So after we got all our GPS data and we put it into ArcGIS, I take the center points, and that's what this little triangle or green dot you see right there is. And so I would put that in there with all the layers for, we use LiDAR layer, a Landsat layer, a MODIS layer, and an NDVI, which is Normalized Difference Variation Index. And so I'd zoom all the way in as close as I could to figure out exactly where that center point was. And so what I would do is I take, I don't know if you can see it's too good from far away, but there's a pixel here, a pixel here, a pixel here, and a pixel there. And so I would take a 4-pixel average of the 4 closest to the thing, making a perfect square. And then I do a 9-pixel average with the center spot right in the middle of the 9, so 3 above, 3 in line, and then 3 below. And so, and then for MODIS, the pixels didn't get far enough down. They were only 1 kilometer squares, so I just used whatever pixel it happened to be in, because these ones got down to 30 meters, and that's what our sites were, so it's kind of a good number for us. So some results. LiDAR was our best correlation we had for the 9-pixel average, was the LiDAR 9-pixel one, which was an R-squared value of 0.78, and then a really low p-value, so it was pretty good, significant data there. It didn't really surprise us. We were kind of hoping that we'd get a good correlation like this. The other ones, however, did not go as planned. So this is NDVI, which is kind of a widely used LAI product. And as you can see here, there's really no correlation. We had a 0.07 R-squared value, and that was really surprising to us. And then like MODIS and Landsat also were even worse than this one. They were like 0.03 and 0.04, I think. And so then what does all this mean? The data showed that the LiDAR version 1 layer that we had to use had a strong correlation of 0.78. But surprisingly, the NDVI one did not, and what's really surprising because there's a white paper that was published in 1997 that their data showed that they had a 0.97 R-squared value, which is way different than ours. Theirs is way up here. Ours is way down here. You can't even see it. So that was really surprising, and I don't know why why theirs is so high and ours is so low. That's like kind of future research someone probably needs to go down and look at. But then people just kind of use NDVI because of this paper as a LAI product they just use. So our research kind of shows that maybe they should not use it at least for our area. So maybe they should look at LiDAR or do some more research to see if our data is correct. And so conclusions, LiDAR version 1 was the only one that had a good R-squared value, even of 0.1. I mean, nothing had anything over that. So, and then NDVI had no good correlation. And then Landsat and MODIS was less than 0.05. And like I said, MODIS had that really big pixel value. So, I mean, that's one possible reason why it wasn't very good. So, and then references and questions.
The white paper you mentioned, were they testing at the same
They were in Montana, in a montane forest, which, I mean, shouldn't be too much different than what we tested ours in. So I don't know what time of year they tested theirs at, though. So we did ours in the summer. All our laters are from the summer, so we still should have had some kind of correlation. But, yeah.
I'm curious about your survey of the literature, because if this is true in lots of different areas and there's this lack of correlation, how wrong are how many papers?
Oh, I don't know, and that's something people should see why ours is so low and theirs was so high. Why is there a difference there? So, but yeah, like people kind of widely use NDVI because of that paper. So,
Do a lot of people use the LiDAR?
I don't know if people use LiDAR as much, but yeah, I do know that the white paper is widely used, so they use the NDVI. So, but our data showed that LiDAR should be used instead of NDVI, at least for our area. So, yeah.
Great, thank you.
All right. There you go. That might be
Thank
more
you.
helpful.
Our next presenter is Brett Lindgren. His mentor is David Barnhart with Instar and CZO. The title of his presentation is Does Site Physiography and Canopy Type Influence Incident Solar Radiation at the Forest Floor?
How's it going? So I'm Brett. Like she said, we're going to talk about solar radiation and tree canopies today. A few acknowledgments. I'd like to thank my mentor, Dave, for all his time and knowledge, and also my family for their support. So a little background why we're doing this. Most high elevation creek catchment waters is inputted from snowpack and melting snowpack during the springtime. Also, watershed rights in Colorado are very important to predict because it's such tight regulations. Forest canopy structure strongly influences solar radiation transmission. So aspect of the slopes, tree species, canopy type, canopy structure, all go into playing a role in this. And then to measure all this, we're using photosynthetic active radiation sensors, which is the actual particle of light that plants are going to use to create their photosynthesis. So we're not measuring the actual wavelength, we only care about the particles. And so our site we took is Gordon Gulch, or the lower Gordon Gulch, which is about 15 miles west of Boulder. We split it up into 4 sites. Site 1, you can see, is kind of lower on the ridge line, closer to the creek that comes through the middle. The creek is only active during the spring. It does dry up after the snowpack has melted. Site 2 is up on the ridge line on the south-facing side, and it's very sparsely populated with very few trees. Site 3 and 4 are on the north-facing, which is a whole different type of canopy. It is very dense. It's all lodgepole pines compared to the south side, which is mostly ponderosas. With the 4 sites, we split them up into 2 transects each that we label north-south and east-west transects. Some of them are a little crooked, but we still just call them east and west, north and south. So after we had our transects, we measured them out, most measured between 25 and 30 meters. And then we distributed 7 PAR sensors, which is that up on the top right, evenly through, or as even as we could throughout the transect. Some of them got tricky. We only had about 6-7 meter cords, so it was hard to get them very even. They're all hooked up to these data loggers, and due to time restraints, data was collected for 3 days and then downloaded, and then the transect was switched to the next direction. And like you've heard about, this is a hemispherical picture camera to characterize the canopy and the leaf area index. So our hypotheses to start is that the east-west transects will have a lower coefficient of variation, which is just the standard deviation divided by the mean of the PAR readings, than the north-south transect. Also, the south-facing slope sensors will have a higher coefficient of variation in PAR readings than the sensors on the north-facing slope. And Plots with a higher leaf area index, or LAI, will have a lower coefficient of variation in their active radiation readings. And within a site, variation among sensors isn't as important as what type of canopy they're under. We'll get to that soon. So, here you can see this is site 1, 2, 3, 4. This is purely raw data. This is what the sensors read. This is what they put out. It does come out in micromoles per meter squared per second, which is what we use to find the averages and find the coefficient of variation. As you can see, site 1, I kind of compare it as my standard site. It has a lot of sunny spots. It also has a lot of shade. You can, anytime it goes up and down, full sun is close to 2,000 micromoles per second. So you can see site 1 does vary quite a bit. Site 2 is in very little canopy, as you can see from its Hemi picture. This is evident because it stays way up there at 2,000 almost all day. Site 3 and 4 in the different canopies, they do have the up, down, which is consistent with the sunshade cycle that they go through constantly all day long. So this is showing the sensor-to-sensor variation in between each transect. You can see on site 4, each sensor every day was reading very different from the day previously. That's why these boxes are so long. The sensors themselves do have little coefficient of variation, but within their own right, they vary a lot. Site 3, with the really small boxes and high coefficient of variation, that means these sensors were varying from day to day for the coefficient of variation, but from their actual readings were not. So for 3 straight days, these sensors saw the exact same light over that course of the 3 days. Site 2, in a lot of sun, not a lot of coefficient of variation or sensor variability. Site 1 is just kind of the normal in the middle one. So our transect and slope differences and coefficient of variation, we did expect to see that the east-west sensors were going to have more or less of a coefficient of variation. You can see that it doesn't really matter, the transect. It's more what site and what kind of canopy it is under. Even though site 3 and 4 do end up having more variation, it's more due to the canopy than how the transects are lined up. The north, we thought was going to have a lower variation, and it actually happened to have a higher. And we think this, just when I started this and I had my hypothesis, I didn't take into account that sunshade cycle all day. I was just thinking daytime, nighttime, daytime, nighttime. And then leaf area index compared to the coefficient of variation. You can see we have an R-squared value of 0.8, so they are correlated pretty much hand in hand. If we had more sites and more time, you could see this trend would just keep going up as LAI went up or down. Site 2 is at the bottom for leaf area index. It's also at the lowest variation. And transversely, site 3 is the highest variation and also the highest leaf area index. So our key findings, the north-facing sites have more variation than the south-facing sites, which is completely opposite of what we first predicted. Same with this one. East-west transects do not have a lower coefficient of variation than the north-south. This one didn't really matter as much at the end. There is still a little bit of a correlation, but not as much as how the transects are set up than what their canopy looks like. Leaf area index is pretty much the main role of solar transmission through forest canopies. More leaf area, and there's more variation, and it also keeps the radiation from penetrating. Individual sensor readings are more influenced by leaf area index than where they are in their transects. So every north sensor, no matter what, they're more influenced by what they're under than that they're the most north sensors throughout. And then, any questions?
The correlation is with LAI and seedling regeneration in your site specifically.
That's
Deal with the higher one, different regeneration patterns.
That's a good question. I'm not sure what seedling regeneration patterns are.
New trees.
Oh, I would guess that with higher LAI, they would actually grow slower because they're not getting the radiation they need. Unless, maybe on the south-facing site, because there is really low, obviously that site is probably growing a lot faster in the ponderosas than the lodgepoles, which are, they're fighting for the top of the canopy. So that'd be my idea though. Yeah. All right.
Questions?
Perfect.
Thank you. Our next presenter is Scott Wesley Bean. His mentor is Noah Fierer and Tess Brewer, and he'll be presenting on diversity of spore-forming bacteria in soil.
Hi, everyone. All right. So I'm going to move away from trees and LAI and go into something totally different and talk about soil bacteria. I want to take a moment to thank my mentors. They've been awesome this summer. Noah Fierer and Tess Brewer for all of their support this summer, and everyone at Noah's lab for all of their help in making this project possible. And thank you, everybody, for being here. So what if we had the ability to morph into a secondary form where we could resist hot temperatures and cold temperatures? Well, this is somewhat similar to how some bacteria work. In the face of some incredibly difficult and extreme environmental conditions, they will change into a secondary physical form called an endospore. The picture on the right you're seeing, or excuse me, the left that you're seeing is a cell that's in the process of forming an endospore. And then on the right is the end result. This endospore is extremely resistant to temperature changes, to physical desiccation, or excuse me, to desiccation as in drying out, and solar radiation. A classic example of a bacteria that forms an endospore is actually Clostridium botulinum. And their normal environment, they really like low oxygen environments. So when they're in a can and they're not actually sterilized correctly and not removed from that canned goods, they'll actually start growing again. So this interesting category of soil bacteria, or bacteria in general, brought us to our research question, which is, what do the communities of spore-forming bacteria look like in soil? It's a category of bacteria that hasn't been studied in soil ecology a whole lot. So to answer this question, we chose 2 location sites in Gordon Gulch and to collect soil from, the north-facing slope and the south-facing slope. The north-facing slope being environmentally different from the south-facing, in that there's a lot more trees and shade, and on the south-facing slope, you're seeing a lot more solar radiation and a lot less vegetation on that side. So we put both of those samples and divided them up into 2 groups. One that underwent an ethanol treatment, and what this ethanol treatment would do was essentially wipe out a large portion of the community that was not resistant to ethanol, and it would isolate for those endospores or spore formers that we're interested in. And then we'd also have our control samples with the original community that we'd have that to compare to. So in this experiment, we really were not interested in any sort of DNA or genetic information that was from dead cells. When you have soil, there is some free-floating DNA in it that's from cells that have died, but we went ahead and removed that DNA before moving forward and just had the living bacteria cells. From that point, we extracted the DNA from the bacteria, and we used that information to identify the species and the abundance in those samples. So how diverse exactly are these samples? And by diversity, what I mean is simply the number of species, a different number of species in all of these samples. On the x-axis of this figure, you're seeing whether or not they are from the north or south-facing slope. And the color differences indicate whether or not it underwent the ethanol treatment or was a control. So the main trend that I wanted to point out in this figure is simply that the ethanol treatment was successful in lowering the diversity of these of the samples. You'll see that the controls are a lot higher up, and the ethanol-treated samples are a lot lower in diversity. So it makes sense since we're wiping out a large fraction of that community. So yes, it's interesting that there are differences in diversity as a result of the ethanol treatment, but who exactly are the spore formers in these samples? So to determine this, we determined who exactly was more abundant in the ethanol-treated samples versus the controls. So on this y-axis, you're seeing a list of the different bacteria that were much more abundant in those samples. And then, of course, the relative abundance, or how populated they were in that community. And again, whether or not they were in the ethanol or control sample. So I really wanted to dive into how exactly these bacterial species survive the ethanol treatment. So I went ahead and looked at some specific categories or phylum of bacteria, and the first being firmicutes. And as it turns out, firmicutes do in fact form endospores, so it's not entirely surprising that we're seeing them in those samples. And then actinobacteria, which seem to kind of dominate these ethanol-treated samples. Turns out that they actually form spores as well, but they have other characteristics that might actually put them above firmicutes. They have a really waxy coat, and they also have a high lipid content in their cell walls, or a high fat content. And what that does is it makes it that much harder for the ethanol to penetrate their cell wall and to actually kill them. So this is where things get pretty interesting. We didn't expect to see proteobacteria in these samples, especially because not many of them form spores. But looking further into it, I found that there were two in particular, Xanthinobacterium and Rubivivax, that form symbiotic relationships with fungus. And what that means is that they live closely together, and they help each other out, essentially. They actually reside within the fungal spores of the fungus. So likely what happened was when we collected that soil, we also collected those fungal spores, and that's why when we put it through the ethanol treatment, we still had proteobacteria DNA. There's also another group called Atherobacter that forms a different type of spore. It's more like a fruiting body than a spore, but it's called a myxospore. So I wanted to go and split up the data into north versus south and ethanol versus controls. So what we're seeing here is still relative abundance, but the different squares are representing exactly how abundant they are in each of these samples. So blue being a low abundance, yellow moderate, and green a high abundance. So here's our lovely list of spore formers that we found already. And then whether or not they are from the south or north facing slopes, and then whether or not they underwent that treatment of ethanol. So the same trend that we saw in the last figure is represented here as well. You'll see that the controls are mostly blue, so there wasn't a whole lot of those spore formers in those samples. But in the ethanol-treated samples, we're seeing a much higher abundance. Actinobacteria being the most abundant in these samples, followed by proteobacteria, and then firmicutes. Interestingly, there seem to be some species that were more abundant in one slope over the other. One being Mycobacterium, the actinobacteria, that seemed to be more abundant in the north-facing slope, and then Methylocystaceae, which was a bit more abundant in the south-facing slope. So this might be accounted for different soil conditions, but it's not clear why exactly they're more abundant in one over the other. So in conclusion, not all of the spore formers in soil are going to be endospores, and this was actually contrary to what I was expecting. Our ethanol treatment was, in fact, successful in isolating spore formers, but it also generally are selected for bacteria that have different strategies of survival and resistance characteristics. So some future research, I definitely want to look further into Methylocystaceae and Rhodoplanus, because they're two proteobacteria that I couldn't find why exactly they would be able to resist that ethanol treatment. And I'd also be interested to look in why there are differences in relative abundance from one slope to the other, and possibly what the conditions are that account for that difference. Any questions? So that diagram makes it seem like we kind of just remove it physically, but that's actually not the case. We add a DNA binding dye called propydium monoazide, or PMA, and it's a light activated dye. So essentially you'll put it in those soil solutions that you saw, and it'll bind to that DNA that's free floating, and it can't bind to the DNA within the cells themselves. And then when you put them through a process called PCR, you copy those genes to amplify them, it won't go through that process and it won't copy those genes, so they won't be part of the sequencing data. So it won't be part of the data that we need. It's only going to represent the living cells that we're interested in.
Thank you. Our next presenter is Taylor Schoenfeld, and his mentors were Greg Tucker and Charles Shoby. He'll be presenting on processes affecting spatial variability in sediment size at Chalk Creek, Colorado.
So this summer I looked at a debris flow near Chalk Creek that comes down off of the chalk cliffs and enters the channel at Chalk Creek. And it's not actually chalk, it's this hydrothermally altered granite that's really weak and brittle and breaks apart really easily. So my research question is, how does a point source of sediment supplied by a debris flow affect fluvial sediment size or grain size in the river? And a debris flow is this flow of sediment. When it rains a lot, it comes rolling down the hillside into the channel. So it was a really cool natural experiment that I got to take advantage of and look at how this debris flow affects the grain size in the stream. And the reason this research is important is sand is actually an $80 billion a year industry because it's used in construction, concrete, and in fracking. So we lose a lot of sediment and sand from rivers flowing out into oceans. One of the processes I looked at was the hill slope supply from the debris flow, where the grains either come down slowly through erosion on a day-to-day basis or in these big debris flow events. Another process that affects grain size is selective transport, where the small grains are easily entrained and transported further downstream. I have a video of selective transport. So you can see the smaller grains getting washed away and the larger grains are remaining. And then another process that affects grain size is abrasion, where grains at the source of a stream start out really big and angular, and as they tumble downstream, they abrade and become smaller and more well-rounded. So I took samples at 7 sites on Chalk Creek, 5 sites in the channel, and then 2 on the debris flow. So I laid out a tape measure about 20 meters, and every 10 centimeters, I picked up a grain without looking and measured the intermediate axis. I used a gravelometer, which is this plate with different size holes, so each grain fit into a size category. So this is one of the sites on the alluvial fan on the debris flow that's entering Chalk Creek there. So this is Chalk Creek flowing down into the Arkansas River, and you can see the debris flow coming down off the chalk cliffs into the channel. And I wanted to see how the debris supplied here from the hill slope affected grain size downstream. And I thought it would only affect the grain sizes downstream, but it turns out it actually affected grain size both upstream and downstream. So where the debris flow comes down and enters the channel, there's a really high slope that we measured with a laser rangefinder, and that creates a higher shear stress to transport larger grain sizes downstream. But it also creates a really shallow slope directly upstream from the debris flow channel confluence. So that allows the sediment to be deposited, and it recharges the carrying capacity of the flow. So even larger grain sizes can be transported further downstream. And I found that anything less than about 11 millimeters is transported immediately away from the fan site further downstream. So this is a chart of all the sites I measured, 200 grains at each site, and the percentage of samples at each site that's finer than a given grain size. So this green line shows the median grain size at each of these sites. And at the site far upstream from the debris flow, we have these really large grains. Then further downstream, just before the debris flow, we have downstream fining, so finer grains. And then these yellow lines are the measurements on the debris flow. So we have this heterogeneous grain size supplied, lots of small grains and large grains. The small grains are swept away downstream, and the grain size jumps back up from the blue line to the red line at the fan. Then slowly, as we go downstream, it starts getting smaller again. The grain sizes fine out. So something we observed is there's a direct correlation between slope, channel slope, and grain size. As the slope increases, so does the grain size, because the smaller grains are more easily transported downstream. So the D50 are the blue dots. That's the median grain size, the 50th percentile at each site. D84 is the 84th percentile, and then D100 is the maximum grain size at each site. So we saw that downstream fining was not consistent here because of the debris flow. But overall, there was a general trend of downstream fining about 8 kilometers downstream from the debris flow. We also found that the point source of hill supply affects the channel morphodynamics, so how the channel flows. The debris flow created a kind of stationary nick point with this high slope downstream and the low slope upstream. And abrasion might not be as dominant a process in grain size, determining grain size, as I initially thought. I think selective transport had a lot more to do with the downstream fining after the debris flow. And I'd like to thank everyone who helped me with this project and the RECCS program. And any questions? Yes.
Is it a concern with those gravelometers? I feel like there is, that's why I'm asking the question, that you preferentially sample larger grain sizes? Because just when you're walking around in a stream bed, you're not going to pick up a sand grain or a silt grain, right? You're going to pick up whatever. So is there a concern about that? And if so, how do you correct for it?
Right. I think there is a concern with that. And what I tried to do was pick up a grain without looking at it. And it's easier if there is like a sandbar, you're going to get some sand. And just whichever the first grain I touched when I reached down. But yeah, that is definitely a concern. And then there are larger grains that won't pass through the gravelometer. So we used a tape measure to measure those. But it was kind of difficult to differentiate between the smaller grains. Yes?
So you said that you saw it was surprising compared to what you first thought. And so what kind of questions do you still have about the system, given that it was surprising?
So I'd kind of like to see between my furthest downstream site and the debris flow, how those grain sizes look, and kind of see where it starts fining out specifically in the channel from the debris flow. I'd also like to compare this to a mountain stream that's more typical, that doesn't have a debris flow, and see how the grain sizes compare. Thank you.
So actually, we have a break. Yeah, yeah. 10 minutes. Yeah, we have a break, and also if you didn't get a list of people and where they show up on the schedule, we have some more of those if you'd like those. We're about 5 minutes ahead, which is great. So we'll take a 10-minute break, and we'll be back here at 5 after. Restrooms are a little bit hard to find, but they are here. And Jennifer, I don't trust my sense of direction. Yeah, I know where they are. They're across the bridge. So if you go out through a door,
I do, but this doesn't look familiar. Don't hurry up. Yeah, all over. Practice a lot when they get up there and let me ask you.
I'm not a Mac person, and I don't know how to make it go into slideshow.
We were at the beginning.
Yeah. Yeah, and that should be. Well, we have a PDF here. I can get it on the system
when you've got a second.
So three things you need to do.
I can go get a thumb if thumb drive. If that's
You
easier.
want me to email it to you?
It's easier to put it on a thumb if it's accessible.
I got it. Okay.
I have it on. There's two right there. What about taking one of these? Just grab one of those.
Wow. So is
Going to be Mila next.
yeah.
I think I've got all the others. I'm going to scan them a little more
So
carefully.
Amanda show up, she's here. Yep. Okay, so do me a favor.
I'm going to go grab it too. I'll be right back.
In the future, always bring a USB.
Okay. Well, I just nobody else had to use it.
No, they didn't have to, just
Yeah,
backup.
I didn't.
Yeah, no, I'm just thinking, in the future, it's a good thing to have. You never know. Stuff happens. So, we got this. No, that's what I mean. I don't know.
Yeah, there's a little, I just used the minus button.
Yeah.
Yellow one,
Yeah.
and then I went, because in the thing, this one, so you just click your name.
Yeah, I need to read last. Okay. Read that upside down.
Data traveler.
Okay, I don't want that one. All right, so. Do you want to see this, or do you want to go through it? Right. Do you want, yeah, so just look for your name and,
Yeah, no, I'm talking about what's still up there, just
It'll show up here.
Yeah.
minimize my.
Yeah, okay. So let's go get yours going here. So here's,
Yeah.
And no one's using a laser pointer, so they're here. I don't know if you need them.
I think we're nervous
I
that
think
we're
it-
gonna shake.
Okay, well, it's up to you. My
Yeah.
yeah. It's great. It's not enough. Do you want me to move this table all the way too? Is that kind of irritating having this here?
I'm not gonna walk around.
It really does.
You live from here, just escape, and then.
Oh, you
It's,
got it.
great.
Halo.
Okay, let me try.
It's
Awesome.
Yeah. And I'm gonna sit over here, Kayla, and do 4 minutes. I'll hold this up, and then you have 1 minute left for your questions. I know. I know. You're perfect. That's why I said that. Then no one's gonna.
Yeah. I knew I wasn't going to go long. But I was like, I was up there, I was like, I don't even know what time it's not.
No, I mean, if you just sit with you, okay,
All I gotta go back to the rank building. I gotta turn
Your PowerPoint. I want to make sure
Good. All right. Perfect timing. Great. So our next presentation is Michaela Branham, and her mentor is Jimmy McCutcheon. Her presentation is on the study she did about nitrogen or phosphorus, the limiting factor on algal growth in Colorado lakes.
Okay, so welcome back from the break. This stops freaking out. Okay, so like Suzanne said, I did nitrogen and phosphorus, the limiting factor on algal growth in Colorado lakes. Basically, I'll just start with some vocabulary. I worked with Jimmy McCutcheon this summer, who is with the Department of Limnology, and limnology is the study of inland waters. So that can do with lakes or rivers, and this summer I chose to work with lakes. So we looked at the limiting factor, and that is the nutrient in shortest supply relative to the needs for growth. So eutrophication is the nutrient enrichment of bodies of water such as lakes, and the growth of algae is limited by the availability of nutrients such as nitrogen and phosphorus. Under some other special occasions there can be iron too, but for the purpose of this study this summer we did nitrogen and phosphorus. So purpose was to decide whether or not nitrogen or phosphorus was limiting to algal growth. So here I've got some pictures of my methods. Okay, so first the boat that I got to go out on the lake, I would take a 4-liter bottle and I would fill it up with surface water from 1 to 2 meters. And then second picture, I would bring it back to the lab and take 4 1-liter containers. And I would have a control. I would add nitrogen to one, I would add phosphorus to one, and then I would add both to one. So I added 1 milliliter of each nutrient to the bottle, depending on the label. And then I would take it up to a greenhouse, and I would put it in a waiting pool that was at about 24 degrees Celsius. And I would let it sit there for 3 to 4 days. I would then bring it back down to the lab, and I would filter it. And I would take the filters that I used for that, and I would stick them into test tubes, as you can see right here. Not anymore. But and then I would add 90% ethanol, 10 milliliters of that to each test tube, and I would heat it up to 78 degrees Celsius because that's the boiling point for ethanol, and that would extract the chlorophyll. And then I would usually stick that into a freezer for 24 hours, and the next day I would come in and I would run the chlorophyll samples through a spectrometer, which measures light intensity. As you can see, it gets really green. You can definitely tell there's chlorophyll in there. That was a nitrogen plus phosphorus sample. So our results, our hypothesis was that phosphorus was going to be the limiting factor because in past studies that's what the result has been. And usually if you go to the ocean, you'll find that nitrogen is the limiting factor. So as you can see from So green, we have nitrogen, blue, we have phosphorus, and orange, we have nitrogen and phosphorus. So as you can see, phosphorus was overwhelmingly the limiting factor in most of the lakes, especially in the mountains. We split it up between mountains, foothills, and plains, and I did 7 different lakes over a 10-day period of time. On the bottom, we have the location, and on y-axis, we have chlorophyll, the percent above control. So with the spectrometer, we measured how much chlorophyll was above the control. Here we have dissolved inorganic nitrogen and versus the chlorophyll percent above control. So dissolved inorganic nitrogen is nitrates plus ammonia, which you can see in the nitrogen cycle. And that really had no correlation with the chlorophyll percent above control. But when we get down to phosphorus, which I believe is the amount of phosphorus absorbed by the filter that we use, you can definitely see that there's a correlation, an exponential decrease between total dissolved phosphorus and chlorophyll, which percent above control. So discussion in past, so I'll go back. Mountains 3 in the past, in a study done by Don Morris, nitrogen actually was the limiting factor, but here I found that phosphorus was the limiting factor, so that could be based on the time of year that we took the sample, or temperature. Number of factors could have played a role in that. Then we also decided that lakes with high initial nutrient concentrations tended to show weak responses to nutrient additions compared with lakes with low nutrient concentrations. So our conclusion was that phosphorus out of these lakes in Colorado was the limiting factor. And then, just an observation was that when we added nitrogen and phosphorus to a lake sample, that grew the most algae out of all of them. So the importance and future study, basically why this is important to study in inland waters and the ocean is that too much algae can cause harmful algae blooms, which can kill off fish. It can cause oxygen depletion, and it can also release toxic metals into the air. That's obviously not something we want. So in order to improve this experiment, if I would have had more time this summer, I would have liked to maybe gone to even more lakes, but also with the ones that I had, I could have gone up there multiple times, and I could have taken at least 2 samples each just to compare them to make sure there weren't any errors. Acknowledgement, thank you, Jimmy, for being a very helpful and resourceful mentor and being patient with me. And then also thank you to all of your researchers in your lab. They really helped me out with the steps to complete my project. And then thank you, Jennifer and Leslie, who's not here, and also Beck for helping out with scientific communication and the RECCS program as a whole. Any questions? That was just where we decided to stick the wading pools. That was in the greenhouse above the building I was in. That could be another thing you could do down the future is have 2 different wading pools of 2 different temperatures and see if there's any effect there. Or set the wading pool to the temperature from the lake that you took it from to Woodward. Scott.
How did you extract the chlorophyll?
That was my favorite process. Let me go back to the picture. So you can see a needle right here. So I would take it out with the needle or the syringe from the tube, and I would try not to touch the filter, but I ran it through a centrifuge for 5 minutes at first to get all the chlorophyll to the bottom, and then I would extract it with this syringe. And then right here, there's a filter, and I would unscrew the syringe and screw that on, and then I would very slowly put into a 5 milliliter, forget the term, but it just it goes into the spectrometer, and then I'd run it through. It would do it for me, so I get the numbers. Anyone else?
Thank you. So, William, is there anything we need to do about the... Is there anything that we can do? We don't know. We're going to hope that the thing doesn't keep going on and off. So it looked like maybe, yeah, it looked like when you touched the space bar, it stopped doing it.
I think it's because she had the battery pack right next to the kids.
The battery pack? Oh.
From the microphone.
You think that? You think it was interfering?
Well, it was like, I think it was hitting it.
Hitting the cable when she moved?
Like when she pulled away and stuff.
Okay, so there might be some oddities with the connection with the cable. Yeah, that's weird. Okay, thank you. Great. All good, you think? Okay, fingers crossed. Our next presenter is Shayla Wallace. Her mentor is Brian Harvey, and Shayla will present on tree ring width index relationships with drought in the subalpine forest.
Hi everyone. Again, my name is Shayla Wallace, and today I'll be talking to you about drought in the subalpine forest and what effects it has on these forests. So I'd like to start off by thanking the crew that helped me out this summer, starting with my mentor, of course, Brian J. Harvey, a few of my peers, the whole RECCS and series crew, and of course all the funding sources such as the NSF. So to give a little bit of background about drought in general, we all know that drought affects trees. How they affect these trees and their individual traits is a little less well known, such as their different species, different size class, and things like this. How is that going to differentiate when it is affected by drought? Understanding how different trees are affected differently is really important to understanding the vulnerability of our forests. And you can see over here to your left, we're going to use tree ring analysis with these cores to find out some of these questions. So to start off, I'd like to show you just a little animation to better explain how we went through this process. We have 3 sites here that we chose. We have a warm dry site, which is a little closer here to Boulder. We have a cool dry site, which is up at the Mountain Research Station. And then we have a cool wet site, which is a little closer to Ward. The raindrops are indication on the different precipitation at these sites. And then we are going to see the intermediate size of these trees and the dominant size of these trees and how they differentiate as far as the effect of drought has on all of these. So again, a couple of the questions that we have for this summer is how do climate versus tree growth relationships vary between a couple of gradients, such as the plot and the local climate conditions associated with these different plots, as well as the different tree size classes, the intermediate and the dominant sizes. Our hypotheses for these 2 questions happen to be that the warm dry site, there you saw at the bottom, was going to be the most negatively affected by drought, and the dominant tree size class was going to be the most negatively affected by drought as well. And I'll go in a little later as to why we thought that. So to start, we have a few different types of methods that we use throughout the summer. First, of course, is data collection. What we did for this was, once again, picked our 3 plot sites. There you can see me coring a tree. So we have 60 cores in total, 20 from each site, and then we brought them down a little bit more specifically for species and their different size classes to have a big sample size. We then sanded and mounted all of these tree cores and processed them through a system called Windendro, where you can see here it's counting each individual width of each individual annual tree ring there. Once we were able to get all this information, detrend it, and look at it, cross-date it to make sure for no errors, we were able to analyze all of our data at that point. Now what we did for our study for analyzing it is we did it against what's called a PDSI for the climate portion. What this does is it looks at the precipitation versus the temperature of that year, and then it comes up with a climate that would be for plants. Once again, here's a figure of the different plot sites that we had, the warm dry one, cool dry, and the cool wet. The light area you see up top there in the middle is the Niwot Ridge, to give a little bit more of where we had our plots at. So to start with our results, I'm going to show you each individual graph that we had for each individual plot site that we took from. So as you can see up top here, this is the cool wet site. The RWI over there on the y-axis is what the ring width measurements were. And here at the bottom is the water year PDSI, which is again that temperature and precipitation climate factor. You can see here at the cool wet site that we didn't have a correlation much at all against the PDSI here on the bottom or between the 2 dominant and intermediate size classes. And we'll once again go into that a little further later. We now have the same kind of graph, but with the cool dry site, which is the Mountain Research Station again. Once again, we have a little bit more of a correlation with PDSI, but no correlation whatsoever between the 2 different size classes. And our last plot site, the warm dry one. This is where we really start to see some results. We do see a correlation between the PDSI as well as with each other, the dominant and intermediate size classes. What this means is that these 2 lines are a little bit different from each other compared to the other sites that we saw before. This is just the cool wet site versus the warm dry site to give you guys a little bit more of a visual of the differences between the 2. Now we'll go into all of the dominant versus all the intermediate trees. This is a graph just for all the dominant trees across all the plot sites. As you can see, once again, there's no correlation with the PDSI or between one another. What this means is that dominant trees grow the same throughout all the different plot sites regardless of drought. Then once again, we hit the intermediate trees and we see something completely different. We see that there is a correlation, at least with the warm dry site, looks kind of tan on this guy, with the PDSI as well as with each other. And here's another side-by-side to give you guys a little bit more of a visual. So now we can kind of talk about the results that we just saw. Starting with the warm dry site. This was the one that was most negatively affected by drought, just like we had thought with our hypothesis in the beginning. This is because this site is already had a limited access to water. So without or with drought, it leaves less water even compared to what they already have access to. Then we go to the cool dry site. It was kind of in the middle, a mediator between the three sites, which is pretty expected given where it is on our animation. Now the cool wet site. We did not have a correlation between anything on our graphs. This is because the cool wet site has a very small growing season. What happens up at this site is it is so snowy and cold all year round that it only has about a month out of the summer to grow. So what drought actually does for this site is it could potentially help it with lengthening that growing season. Looking at all the dominant and all the intermediate trees within each plot, we didn't see much of a correlation. Same thing with the dominant trees as we saw in our one graph. We didn't see much of a correlation between one another. But with the intermediate trees, we did see quite a relationship with drought. We thought the opposite, right? When we were looking at our hypothesis earlier, we thought the dominant trees would be most negatively affected by drought. This is because we thought that they were going to be up at the top, had the most access to weather, sun, things like that, more up in the elements. Turns out the intermediate trees were actually the most negatively affected by drought. This could be because the dominant trees actually have more of a root system, which helps them get moisture elsewhere, whereas the intermediate trees do not. So the conclusion or the big picture about this study in particular is that a lot of things matter when we're looking at how drought affects trees. It is important to know not to lump all trees or all plants together when we're looking at climate. So basically, site matters. Where the tree is located matters. The tree size class matters. How big that tree is matters. Effects of drought are different everywhere. This is really important for a global stance to know that with possible warming climate, that we need to look at every area differently. And then continued research, I will actually be continuing with my mentor at CU starting in the fall. And what we've already started to do actually is we're looking at the carbon isotope content and ratios in these individual annual rings that we've already used for this study. Basically, C13 versus C12. C12 is taken in by trees because it's easier to break down because it's smaller. C13 only is taken in if it absolutely has to. So basically, if a tree is drought stressed, it's going to be taking in that C13. So what we're hoping to see in our future research to support what we have done here is that the smaller ring widths means the higher C13 content. And if anybody has any extra questions about that, please come see me. It's really exciting. Thank you. Brett.
Did you, when you were taking your samples, did you restrict to just using like lodgepoles and ponderosas, or did you take samples from like spruces and stuff too?
Yeah, so we did only spruce and fir. We did subalpine and Engelmann. And the reason we did this actually is because the lodgepole pine and ponderosa, they are well known in the research community, and the fir and spruce aren't, which I can kind of see now why, because the rings were tiny on the spruce and fir. But yeah, those were the species we used. Yeah. Go ahead.
Can you tell me how you trended the data? That's sort of the first process that you have to go through before you then have a time series to look at.
Yes. So detrending basically means that when you have a tree sapling, it's growing a lot each year, but then when it gets bigger, it kind of slows down. So what happens is we can't look at each individual raw data for those ring widths and think, oh, it grew a whole bunch here, but then it slowed down, so it must be due to climate. So what happens is we have to detrend that series to make sure that that trend does not show up when we're looking at our data later on. And what did that was the wind dendro system that we used. It kind of did it for us. So that was good. Great. Yeah. Anybody else? Awesome. Thanks, guys. Oh, no, I'm keeping it.
So if you go to this folder, so if you go out of that one, that red one, and then yours would be. I'll introduce you. So you can make that slideshow. Okay. Our next presenter is Ward Fish. His mentor was Imtiaz Rangwala, and he'll be presenting on variability and trends in snow water equivalent and stream flow at the Wind River Indian Reservation in Wyoming.
Hi, so I am Ward Fish. The goal of our project was to study the climate trends in the hydrologic cycle in the Wind River Indian Reservation in Wyoming. So we set out to examine recent historical trends in snow and rain and stream flow in the Wind River Indian Reservation and the surrounding watersheds, which encompass the Wind River Range. Our motivation was to inform the ongoing Wind River Indian Reservation Drought Preparedness Project in the region, which is led by the USGS's North Central Climate Science Center and NOAA, the National Drought Mitigation Center, and NOAA's Physical Science Division. My mentor actually just presented this same study on Tuesday to the in Wyoming to the tribal water management team in Wyoming. So the Wind River Indian Reservation is outlined here in white. This is Wyoming, a map of Wyoming. The Wind River Indian Reservation is here outlined in white. And this right here is the Wind River Range, slightly to the west, but feeds the Wind River Indian Reservation. I should note that the Wind River Range tops out at about just under 14,000 feet. The Wind River Indian Reservation is home to 26,000 inhabitants belonging to the Northern Arapaho and Eastern Shoshone tribes. These people are rely heavily on the natural snowpack reservoirs and the snowmelt from the Wind River Range. In recent years, they've been experiencing severe drought. So let's dive in here. One of the most interesting trends that we noticed was that the temperature in this region has increased by 2 degrees Fahrenheit in the last 67 years. Additionally, in the same period, we noticed that the freezing level, or the snow line, has increased by 500 feet in elevation. This means that the elevation of freezing temperatures has risen by 500 feet for every degree. That means the freezing level went up by 250 feet in elevation. This suggests that the natural reservoirs that these tribes rely on so heavily for irrigation will be diminished and in some cases non-existent. So what we did was we looked at all the available high elevation observations of climate data. We looked at the data compiled by the SNOWTEL network. We looked at the last 35 years worth of precipitation and snow water equivalent, or SWE. And SWE can be best described as the water content in snow. Like if we were to fill this room with all sorts of different kinds of snow, dry and wet snow, and it were to instantaneously melt, how deep would the water be? That's SWE. We examined the proportion of rain and snow in the precipitation, and we used a temperature-based method proposed by Kinzel, 2008. We studied the trends in the rain in snow and SWE, especially the change in timing of snow to rain shift. And then we studied 2015 separately, excuse me. So we chose these 12 snow-tail stations circled in magenta by proximity to the Wind River Range and availability of data. So this chart shows, or this graph shows, the precipitation over the last 35 years in just one station. Due to lack of time here, I can only go into one station. The thick red line represents the average of the current period, and the thick blue line represents the average of the previous period, 1979 to 1999. The yellow line represents 2015. So as you can see, there's not a huge change in precipitation from the previous period to the current period. But what we did find was a significant difference in snowpack from the previous period to the current period. Same thing as the last slide. The red thick line represents the current period, average of the current period, and the blue lines represent the previous period. The yellow line is 2015. As we can see, it's getting worse, even worse than the current period. So this is important to note that while there was the same amount of precipitation at these stations, the Wind River Indian Reservation still experiences severe drought. So they're getting the same amount of precipitation, and that was our question, is why are they experiencing this drought? This chart explains that the change in the number of days between 2015 and the long-term mean. The number of days between when the rain shifted to, from when the snow shifted to rain. The precipitation is coming as rain rather than snow. So for 2015, however, the snow to rain shift occurred 3 to 7 weeks earlier in spring, which is pretty huge. So for 2015, in this chart, we ranked all the stations. And as you can see, the total precipitation, we ranked total precipitation and maximum SWE. As you can see, the total precipitation is around the median. This is over 30 years. The 15 is obviously the median. And as you can see, the precipitation is around the median, and the SWE is significantly less in every station except for Kerwin there. But this is huge. As I said earlier, that they rely on the snowpack, and the water doesn't really do them any good if it comes in January or February like we've been seeing. So in conclusion, between 1948 and 2015, the Wind River region has warmed by 2 degrees Fahrenheit, and the atmospheric freezing level, or snow line, has moved up in elevation by 500 feet. The period since 2000 has been relatively drier in the region compared to the earlier 2 decades. However, there's no clear consensus between the stations and an earlier shift in the timing of rain during that period. For 2015, however, the rain shift occurred 3 to 7 weeks earlier in spring. And while I didn't talk about any stream gauge information or data today, we did all those same analyses for stream gauge. I just don't have time to go into it. Stream gauges generally show that the day when half the water year discharge occurred has shifted earlier in recent decades by a few days to a week. And in 2015 especially, the shift was also earlier by a week. And I'd like to thank Dr. Candida Devis, Dr. Linyan Chang, Dr. Joseph Barsugli, and to RECCS and Series for all the resources, especially to my mentor, Dr. Imtiaz Rangwala. He's the most patient man I've ever met, and I made sure to test his patience weekly. So, any questions? Hobbs Park.
What's your hypothesis on that?
Which one now?
Kerwin.
Kerwin. It was in. We went in ascending elevation, and at first we thought, well, maybe elevation had something to do with it. I honestly don't know, because they're both on the eastern side of the Wind River Range, all of those snow-tail stations are generally the same, except for within 1,000 feet of elevation. So we just don't know. It's further study. I should also mention that while the Wind River Range was upstream and to the west of the snow-tail stations that we picked, those were the highest snow-tail stations. The one I showed there was Hobbs Park, was at 10,000 feet. As I said, the Wind River Range peaks about 14,000 feet. So a lot of this precipitation that's affecting those snow-tail stations was coming from snowpack that we had no way to analyze. That's another study for another day. Yes.
So yesterday at the poster session, you mentioned some of the reasons that this is important and some of the ways the reservation might be using this information. Do you remember what those are? Can you describe how this is applied?
So yeah,
Decisions people have to make?
yeah, it is important. So they have, there's a lot of politics involved. The Bureau of Land Management, Bureau of Reclamation, Bureau of Indian Affairs, Department of Agriculture. There's so many acronyms involved in this, and it's a huge project. It's been going on for a couple years now, and will be going on after I leave, obviously. But my mentor was just up there and talking to the drought preparedness mitigation team. And basically, the reservoirs are in disrepair. They're leaking, but they've been working for the last several years. The problem is, since the water's coming sooner, and sometimes it's not coming at all. Like last year, they released in Washakie Reservoir, they released a lot of water in anticipation of the runoff that usually comes every year at that time. Well, as I said, the freezing levels rose by 500 feet in recent years, and that snowpack doesn't exist anymore. So, yeah, they need more reservoirs to catch the rain and use the rain when it's coming instead of relying on these natural reservoirs. Does that answer your question?
Yeah.
Okay.
Thank you.
Oh, yeah. I don't want this.
Thank you. Our next presenter is Amanda Espinosa Martinez. Her mentor is Michael Gusev, and Amanda will be presenting on stream water temperature changes during summer along the Gordon Gulch Creek.
Okay, so like she mentioned before, my name is Amanda, and first off, I'd like to thank everyone who's helped me throughout this summer, all my mentor, Michael, the RECCS staff. Everyone's been great and super supportive because this has been quite the adventure. So stream water temperature in the Gordon Gulch Creek for the summer of 2015 is what I spent my time studying this summer. So the real world importance, stream temperature is one of the most important aspects of an aquatic ecosystem. Fish, for example, they have thermal tolerances, so they need to be in the right environment for their safety, it's critical. And another important factor is dissolved oxygen, which is also controlled by water temperature, which once again links to fish and other biotic inhabitants. They kind of need to be in the right environment to have the right amount of dissolved oxygen. Stream temperature dynamics are driven by heat exchanges and other atmospheric inputs. So here's a graphic showing atmospheric inputs. The ones we concentrated most on was solar, groundwater flux, and air conduction. So there's plenty more that we could research, which is probably a future study. So here's a map of the Gordon Gulch Creek, which showcases where we studied. Site A is red, and it's going to correlate with the graphs you're going to see. Site, which was at 0M, which is the furthest up. Site B is green at 500 meters, and Site C is blue, which is at 1,000 meters, and that was the furthest downstream. Each of them were 500 meters apart, so there's that. What we used to collect the data was the onset HOBO water level and temperature data logger, which collects data every 15 minutes. So at the end of one day, I have 96 to look at, which is lots of fun. And like I mentioned earlier, this is previously collected data. So Patrick Hendrickson and Joey Gamora collected this for me last year. So that's pretty nice, considering they didn't know they were doing that. The data loggers were deployed from June to August 2015, and they're still in the field actually now. So right here, this is the spaghetti of everything. This is like a big mess. This is the whole plate. So as you can tell here, site A, which is the red one, is pretty consistent. It doesn't change too much. Site B, which is the green one, at 500 meters, it changes drastically throughout the summer. And then site C, which is the blue, it kind of gets washed away in the later half of the summer. But the importance here is that at one point, site C is actually warmer than site B, and they switch about midsummer. They're all super diverse, and they all have their own patterns, as you can tell. So instead of the entire thing, here's just one bite of everything. So this is one day, which was July 4th. And as you can tell, once again, site A is really consistent, still in its range. Site B, which is the green one, if you can kind of tell, it does the same thing it does in the whole one, like overall, where site C is warmer than site B, and they alternate. So it does the same thing on a daily basis, where they have peak levels, or peak times, where they're warmer than one another. Site A temperature is very consistent, whereas the daily maximum temperatures at site B generally are higher. So on the left is the minimum daily temperature stream, or daily stream temperature. And on the left, right, this one right here, that was the max. So the y-axis is from 6 to 21 degrees Celsius, and on the x-axis is the days. So as you can tell, once again, the red's pretty consistent. It stays in its own level, and I like that. It makes things a lot easier. The blue, or the site B, the green one, once again, it changes the most. It's the most dynamic. And site C, which is the blue, sorry, I didn't change the colors. Site red. Once again, it's kind of dynamic, and it starts off, there's times where it's actually harder than site B. So another thing we looked at was the difference in daily average temperature between the sites. So the blue is between sites A and B, so 0 to 500 meters. And as you can tell, it's always increasing. That's because site A, it's pretty consistent, so it's always going to increase from site B. And the yellow is from 500 to 1,000 meters, which is from site B to site C. And as you can tell, it's always less than a 1 degree difference, which is pretty surprising to me because it always looks so different when I was looking at the entire thing. And like I mentioned earlier, later in the summer, they actually switched over. So some of the reasons this is because site A is actually a location where the stream re-emerges from an underground water flow path. So it's really fresh in a sense. You know, it doesn't have time to interact with the environment as much as site B, for example. Site B experiences the most atmospheric heat exchanges just because it's at the optimal place to receive solar radiation, and there's not much shading there. So of course it's going to be a little bit warmer. From site B to site C, there's actually a lot of cooling going on, and from what I've been told by my mentors, there's actually lots of little springs that feed into the stream, which explains the cooling. So, oh, this looks weird. In conclusion, the strong temperature variability suggests that there may be different optimal locations for fish habitats, dissolved oxygen, and other biotic inhabitants. Once again, fish, maybe when they're young, they're better suited to be in a warm environment, but when they grow older, they have to be in a cool environment, so they definitely need to be in their right place. Spatially, stream temperature patterns seem to be very heterogeneous. Each of them are very distinct, in my opinion. You can tell the difference between each of the sites, and the heterogeneity is likely caused by atmospheric input and groundwater influxes. Thank you. That's a question. It was I could go back real quick. It's right around the 9 to 10-degree range. So probably about a median of like 10. So it's fairly cool.
For fish, so for trout, big industry, what's optimal for, do you have any idea what's optimal for?
I don't have like specific information. That's definitely one of the future studies we want to do. There's actually 10 different sites just like this. I only use 3, the beginning, middle, and end. So it'd be really interesting to see how it varies and if there's even better specificness for, you know, where certain, yes.
So this is our last presentation, and after that, we hope we will see you next door in the reception. Not the right one? Okay.
Alrighty, hi everyone. My name is Sean Rouse.
You need this. Sean Ross is mentored by Suzanne Anderson, and his presentation is Rock Weathering Observed in Outcrops and Bedrock Exposed by Debris Flows, a preliminary investigation of grandorite weathering in a landscape context.
There we go. Cool. Okay. Yeah. So, my name is Sean. Like she said, I've been working with Suzanne Anderson of Instar. She's also in the geography department at CU. We've been looking at weathering of rock in Boulder Canyon, like Susan mentioned there. Yeah. So we have landscape development, which is largely composed of basically 2 factors. You've got weathering of rock, which is disintegration of the rock by chemical or physical processes into smaller pieces, and erosion, which is the transportation of those particles, either by mass wasting, as we can see in these photos, which is where you get a bunch of that material moving at once, or in fluvial processes, which is moved by a river, like Taylor was talking about earlier. Yeah, now weathering is not terribly well understood, like as far as the specific processes go in certain places or their rates. So we decided to take a look at that more closely. Now the settings are pretty fantastic for the 2 questions that we were exploring. As far as Boulder Canyon goes, its method of formation is really interesting. I always figured it was just all down cut at once along the entire length. But it's actually formed by what we call a nick zone, which is a slightly steepened area of the channel that began at the range front, which is slightly off screen here. I had a better graphic, but unfortunately that's in my PowerPoint that did not get loaded. But yeah, so at the range front, there's a contact between the hard, like Susan mentioned, granodiorite of the mountain core in this area, really hard, basically granite, close enough for our purposes. And yeah, there's a contact there between that and the softer sedimentary rocks present at the range front and east of there. And that contact, once Boulder Creek cut down to that, the softer rocks of the sedimentary out east, they downcut more quickly, which created a steepened nick zone in the channel, which began migrating westward up into the mountain front. As of now, that nick zone has migrated all the way from the range front to right up in this region right here, the BCO3, which you can't even read on that graphic, unfortunately, is approximately where Boulder Falls is. Kind of gives you a little more spatial context there. So that allows us to explore. So the way rock weathers as it's exposed at the surface, that's expected to increase with time that the rock is exposed at the surface. So the rocks exposed in near the mouth of Boulder Canyon have been exposed for more time. We don't know exactly how much than rocks at the current position of the nick zone. Another question that we wanted to ask was, wanted to explore anyway, was how rock weathers when it's buried under a thin layer of soil, we would expect that to increase because the soil is holding water with active ingredients, I guess, if you will, against the face of the rock, which would speed up its weathering. Methods. So at each of these study sites, I had a better graphic of this. Unfortunately, that's not present now. All of these locations, so the second hypothesis I was just talking about, to explore that, we took advantage of 2 debris flows that were triggered in the 2013 Front Range storm. These, going to this next slide, there we go, cool. So these debris flows exposed a tremendous amount of bedrock that had previously been covered by a thin layer of soil, and we wanted to take advantage of this great exposure to get an idea of how it had weathered subsurface before it was exposed in 2013. So at the debris flow sites, this is in one of those channels, like I was mentioning. Also this picture at the upper left, and also at outcrops, as you can see in this photo right here, we use the same method. We strung a tape measure along the transect to get an idea of spatial reference. And then we used this tool you can see in the upper center. It's called a Schmidt hammer. It's not like you just whack the rock with it and it tells you how hard it is. You basically push it into the surface of the rock, and the black plunger on the front there goes into the body of the hammer. Spring trips it at one point, and that plunger strikes the face of the rock, and depending on how much energy is reabsorbed by the hammer, it tells you how hard it is. With a unitless number we call a rebound number. And so we did that at 0.5 to 1 meter intervals along each of these transects, ranging anywhere from about 5 to 50 meters. And we did those on outcrops in Boulder Canyon, which are these yellow dots here. We chose 7 of those to study the effect of weathering as rock is at the surface, and then we did it at a bunch of sites within the 2 debris flows you can see in that graphic there in the pink. Results. So we got the rebound numbers. I plotted them against distance going up the canyon. This is another view of Boulder Canyon. The mouth is right down here. We've got Boulder Canyon outcrop 3 up there. That's where Boulder Falls is, like I said. So as we proceed up the canyon from the mouth toward the Nick zone, there is a clear trend of rock becoming more hard. So this fits really nicely into our hypothesis. Basically, the rocks near the mouth of the canyon had been exposed for a lot longer than the rocks at the Nick zone. So they're softer, more weathered, like we would expect. And yeah, we did a statistical analysis of this, and it's a statistically viable trend. We unfortunately encountered a confounding factor in the debris flows. As one would expect, they not only uncovered the bedrock, they also scoured down into it. These were tremendously powerful. As you can see from this figure that I adapted from a paper by Suzanne, Bob, and another Anderson cubed, we call that paper. These transported just a tremendous amount of material. So in looking up here at these 2 graphs, there's a definite increase in the strength of the rock as you proceed from the top of the debris flow down to the bottom, because more and more material is being transported as you move down the debris flows. So unfortunately, they scoured away all of the weathering rind on the rocks, so we were not able to test any hypothesis there. But it was a lot of fun checking them out. They're pretty fantastic. Ready? Any questions? Oh yeah, first though, I want to thank Suzanne. She was amazing. She couldn't be here today. But yeah, she was super patient with me, inspiring, and we had a lot of really good conversations as well. Also, all my cohort, you guys are great, and the RECCS folks, my family, and yeah, everybody else. Trevor, thank you, and Bob, you guys are great. Thank you. Questions? Any questions? Yes.
Even though it's perhaps disappointing that you didn't get to use the debris flow tracks as the experiment you were hoping for, it's nonetheless interesting that your data may tell us that they've indeed been more deeply scoured during the 2013 event than at the bottom and at the top.
Yeah, exactly. Yeah, and the statistical analysis on that was a lot stronger than the one that I performed on the Boulder Canyon outcrops as well. It was an R-squared factor of like 0.7 to 0.9, depending on which one. So yeah, it was clear. And also across the debris flow channels themselves, there was a really clear trend that the rock in the center of the channel was a lot harder than the rock at the sides of the channel, where you would expect more scour anyway. So that also supported the hypothesis, well, that alternate hypothesis, I guess you could call it. Yeah. Okay, cool. My other PowerPoint is so much nicer.
All right, so I want to thank everybody for being here. Again, I want to thank all of the mentors who gave of their time and their expertise. And good job, everybody. I'm so pleased to have had